So, you probably came across, if you were doing histology, okay, you probably came across.
There is the one.
So, which one of them works?
Both.
This one works?
Yes.
Okay. In the histology lab, okay, you probably came across a cross-section of a tissue, and I've shown you a diagram of cross-section of what I think is columnar epithelium, basement membrane, and the connective tissue underneath it. If you look at the connective tissue underneath it, it's made up of fibers, collagen, elastin, reticulin, and so on and so forth. It's also made up of cells, mast cells, fibroblasts, macrophages, and maybe some fat cells. But I am not interested in any one of them. Today, our lecture is about the grey background. Can you see the grey background?
Yeah.
Yes.
Better.
Okay, what you might call the ground substance. That's what we're going to talk about. What is in that grey background? It is hyaluronan, proteoglycans, and glycoproteins. And that's what we need to talk about today. So, the extracellular matrix is everything. Okay? The extracellular matrix is made up of the gags and the fibers. Alright? Again, the extracellular matrix is responsible for
Nutrients.
the flowing of nutrients between cells or to cells and away from cells, oxygen, glucose, amino acid, carbon dioxide, hormones, whatever. Okay? Again, the extracellular matrix is made up of structural proteins like collagen and fibronectin and fibrillin and so on, made up of specialized proteins and proteoglycans, or gags. Okay? The gags are actually carbohydrates. They are disaccharide units that are linked together to make these long molecules. This you've already covered in fibrous proteins. Who did this with you? Dr. Hanna? Yeah. So I am not going to be responsible for this, but I just put it in there to tell you about these structural proteins. It's collagen type 1 up to collagen type 4. Again, where it's found and the diseases that will result if you have a defective laying down of the collagen. Okay? Now, I'm interested in this. The glycosaminoglycans, or the carbohydrates that are stuck onto or that are found in the ground substance. Again, it can be located both in the extracellular matrix or the ground substance of the extracellular matrix, and it can be found attached to cells. It is a heteropolysaccharide. In other words, it's made up of two parts. A homopolysaccharide like starch or amylose or amylopectin is just made up of glucose. Okay? This one is a heteropolysaccharide made up of a disaccharide or repeating disaccharide unit. So you have a disaccharide attached to another disaccharide, the same one, attached to another one, and so on and so forth. Which are the gags that are found, or the most famous ones that are found in the ground substance? Hyaluronic acid. What are the sugars that are modified to give you the disaccharide? Again, modified N-acetylglucosamine, N-acetylgalactosamine, and glucuronate and iduronate. Those are the four modified monosaccharides. You'll see the structure in a minute. So two of these will attach to give you a disaccharide, and then you repeat the disaccharide up to 250 times. if you are making chondroitin sulfate. Okay? The GAGs, because they have these, because they...
Same described means the same two units are getting
Yes.
there, not different units.
No. I'll show you in a second. Hold your thought for a minute. So the GAGs, because they have sticking out of it, remember these are monosaccharides, then they become disaccharides. They sticking out of it are carboxyl groups, hydroxyl groups, and then we put sulfur on them as well. So they become highly, highly negative. Okay? They hold on to water and they repel each other. In other words, it's like sliding, they pass each other. That's why they become viscous. Okay? They become viscous as well. So, let's come back to your question. There's a disaccharide. All GAGs are made up of two parts. An acidic sugar attached to an N-acetylated amino sugar. An acidic sugar, normally this would be CH2OH. Because it has become a carboxylic acid, we call it an acidic sugar. This would be a monosaccharide. If I put an NH on it, it becomes an amino sugar. If I attach an acetyl group to that NH, now I have an N-acetylated sugar. So all GAGs are made up of these two things. That's the disaccharide. And now I repeat it. Okay? I can repeat it 40 times, 250 times, 25,000 times. Okay? Again, there's an amino sugar, glucosamine, because this is glucose with an amine group. If I turn this thing here, CH2OH into C double bonded OH, I get glucuronic acid. If I move the glucuronic acid from outside to inside the ring, it's called idiuronic acid. Again, because of the high numbers of carboxyl group, this thing becomes highly negative. Because of the sulfur, it's also highly negative. Alright? And because I acetylate this NH group, I decrease the amount of positivity. That means it's highly negative. Now, let's take, this highly negative thing makes the GAG to hold on to water. Hydrogen bonding here. Now, if you hold on to water, there's my hyaluronic acid in the synovial fluid. If I push on it, if I jump, okay? If I push on it, the water will move out. And then when I relax, the water comes back in. In other words, it's like a shock absorber. You guys drive cars. Okay? It's like a shock absorber that will absorb the shock of me jumping up and down, or the bending of the knee, or whatever. And the same thing here. Again, which are the GAGs you need to know, or must know? Number one, hyaluronic acid, found in cartilage and synovial fluid. Dermatan sulfate and keratan sulfate, found in the skin. Chondroitin sulfate, the most frequently occurring GAG, found in cartilage. And heparan. Heparan and heparin, which is the anticoagulant. That's also a glycosaminoglycan. Again, hyaluronic acid or hyaluronan does not contain any sulfate. And let's go to that. So there's my disaccharide unit for hyaluronic acid. It's made up of glucuronic acid attached to, repeated 25,000 times. Do you need to know the structures? No. Do you need to know which acidic sugar and N-acetylated sugar is found in hyaluronic acid? Yes. Again, No sulfur. Notice no sulfur. All right? Not covalently linked to protein. In other words, hyaluronic acid is not attached to a protein. Covalent. Does it interact with a protein? Yes, it does. I'll show you how in a minute.
That's the picture.
Where is it found? Synovial fluid, articular cartilage. Where do you find this?
Particular.
Yes, you find it lining the epiphysis. Okay? You find it lining the epiphysis of a bone. You find it in the skin. What is vitreous humor?
In the eyes.
Yes. Posterior chamber of the eye, behind the lens, is a small body that is called the vitreous humor and found in loose connective tissue. Next gap. Glucuronic acid attached to N-acetyl galactosamine found in chondroitin sulfate. Repeated 250 times. Found mostly in cartilage, bone, and in the valves of the heart. I changed one thing. I changed the acidic sugar from glucuronic acid to iduronic acid. In other words, I just moved this thing from outside to inside, and that will give me dimethyl sulfate. Same structure, but dimethyl sulfate. The only gag that does not have an acidic sugar is this one here, with galactose, and that would be carotene sulfate. Notice both of these have sulfur. Can you see? All right. This one is repeated 250 times. This carotene can be repeated up to 40 times. In other words, it's shorter than the one on the top. Next gap, heparan. Heparan. Heparan is made up of glucuronic acid attached to N-acetyl glucosamine. Again, sulfated there, there, there. Okay? Where is it found? Again, associated with heparan sulfate proteoglycans that are sticking out of a cell. So you have an integral protein with this heparan thing sticking out. Okay? Also found in the basement membrane and components of the cell surface. If you have a lot and a lot of sulfur on this thing, then you now have heparin, which is more sulfated than heparan. Again, you know heparin is an anticoagulant. Okay, is an anticoagulant. Now, yes, sir.
Can you go back 2 slides? For dermatan, it has same number of units as chondroitin sulfate, right?
Yes, but the sugar is
But
different.
the sugar is different. Do you have to know the number of repeated units in every
No.
one of them?
No. You don't need to know structures, okay? And you don't need to know 250, 25,000, 40, 60, whatever. Okay? Good. Now, how do we make these gags? We make the gags using the ER and the Golgi, because the enzymes to attach these disaccharides one to the other are in the ER and the Golgi. Before I attach the monosaccharides so I can build them up, I need to activate the monosaccharide. So, how do I activate the monosaccharide? By attaching it to UTP, uridine triphosphate. That becomes UDP glucose. I'm using glucose here, that's why I'm saying glucose. If you were using galactose, it will be UDP galactose. All right? Anytime you need to attach one monosaccharide to another monosaccharide, it must be in the form of UDP something. Galactose, glucose, fructose, whatever. So, there's my UDP glucose. What do? I turn it into an acidic sugar. It now becomes UDP glucuronic acid. I'm ready to use it. Now what do I need? I need a piece of protein. Can you see the protein?
Yes.
obviously, the protein, the messenger RNA comes out, ribosomes jump on it, signal sequence, brings it onto an ER. Now the protein is inside the ER. So there's the protein. There's a serine unit. Can you see a serine? And what do I need to do? First, I attach a xylose, galactose, galactose, a xylose, galactose, galactose. All right? I need those three things. A xylose, galactose, galactose, and then N-acetyl glucuronic acid and glucuronic acid and N-acetyl galactosamine. So I must attach these 5 things. And now what do? I start to attach my disaccharide units. Can you see? Yes? All right. One monosaccharide at a time. So I'll put glucuronic acid, N-acetyl galactosamine. Glucuronic acid, N-acetyl glucosamine, so on. But I need this first on the serine. Okay? And that's how I will make this. Once I've reached my 40, 20, 250, or 25,000, I stop. I put this thing into a vesicle from the trans-Golgi, and I send it out into the plasma, to the plasma membrane, and then it goes out. There we are. First I make the core protein, I put my xylose, then I put my galactose, and now I start making, in the intermediate and trans-Golgi, I start to put in my acidic sugar and my N-acetyl galactosamine. I am making chondroitin sulfate. And this thing stays in here until I get my 250 length. Then I put it into a vesicle and I put it outside. Okay. So, remember I needed a piece of protein to make this long thing. We call that whole thing now a proteoglycan. It's a core protein with a long glycosaminoglycan attached to it. It holds on to lots of water, compressive forces, and found in the matrix of cartilage. Again, in the cartilage you will find collagen fibers type 2. Bound to them will be these large gags called aggregans. Okay? Again, I needed a core protein. So, there's the core protein. Can you see the core protein? And I will attach the gag. There's the gag, the chondroitin sulfate, glucuronic acid, and N-acetyl galactosamine, 250 times. But I need this first. The xylose, galactose, galactose, and glucuronic acid, which is attached to a serine. Now, do I make gags on all serines? No. Only ones that have the following sequence. Serine, glycine, any amino acid glycine. That's the serine I will attach or build the gag on. And I must have the xylose, galactose, galactose, and there's glucuronic acid. And now I can start building my gag. Okay? So, aggrecan. This, can you see this leaf looking like structure? Yes? That is aggrecan. It is a protein that's in the middle. That's the core protein. Sticking out of the core protein is chondroitin sulfate and what's the blue one? Keratan sulfate. Keratan is about 40, chondroitin is about 250. Okay? Now, what are these aggrecan things doing? They are attached to hyaluronan. They are attached to hyaluronan. but they are not attached through a covalent bond. They are not attached through a covalent bond. So you might ask, well, how are they attached to hyaluronan? It's like attaching a piece of paper on your, you know, your fridge at home. Your mom puts a little note with a magnet. Yes?
Yes.
That's how it is. It's like you have a magnet.
So my charge.
Okay? By a linking protein. By a linking protein. No charges. By a linking protein called high L adherens. That's how you attach the aggrecan to the hyaluronic acid. And if you have an absence of high L adherens, this leads to failure to attach the aggrecan to the hyaluronic acid, and now that will give you cartilage, defects in the cartilage. And now, I told you what the structure looks like. I told you what's found in it. I told you how it is made. What is its function? Or some of its function. A lot of it is structural. Some of it is for compression. And now let me give you two fancy functions. So there's my cell. I have a cell here. Sticking out of the cell is a heparan sulfate. And the heparan sulfate is made up of parts where there is a lot of sulfur and not so much sulfur. So the green bits are a lot of sulfur, not so much sulfur. Are you with me? Sticking to the green bits is this blue thing. Can you see the blue thing? The blue thing is called antithrombin. And what does the antithrombin do? It holds onto factor 10A. How many of you have done the clotting cascade? We got one person in the room. Two. Okay? The clotting cascade is a series of proteins that need to be activated up to the point of fibrinogen becoming fibrin. Prothrombin will convert fibrinogen to fibrin, then fibrin will form a clot. Factor 10A, okay, is one of those proteins. So what does factor 10A do? It binds to antithrombin. And now you can picture this maybe on an endothelial cell. And if it is bound here, it will not cause any clotting.